Howdy, and welcome to the module on protein structure. In the last module, we talked about primary structure, which is just another way of saying protein sequence. In this module, we'll be talking about three-dimensional structure and the hierarchy from primary to secondary to tertiary, and in some cases, proteins acquire a quaternary structure. So we will be looking at those higher order structure levels in this module. The big questions that we're going to answer when we get to the end of all of these videos are shown here. What non-covalent interactions stabilize protein structure? You know these. These are the non-covalent interactions we've been talking about all semester. What role does the amino acid sequence or primary structure play in the secondary, tertiary, and quaternary structures? What are the elements of secondary structure? What are we talking about when we say secondary structure in proteins, and how are they formed? And how does secondary structure assemble to form tertiary structure, that next level in the hierarchy of protein structure? How do protein subunits, and remember subunits means separate polypeptide chains, interact at the quaternary level of protein structure? And how do polypeptides fold into their three-dimensional protein structures? So that's the order that we'll be going through in this module. There is a video link on eCampus that takes you to the state-of-the-art of deducing the structure of a protein based on its sequence. This is what's going on today in order to figure out, based on other proteins with known structure, how your protein is probably structured. So I encourage you to view that video. All right, so this is the outline of the hierarchy of protein structure. Before we get to the outline of primary, secondary, tertiary, and quaternary, let's group proteins into two categories. Globular proteins, which are soluble and somewhat spherical. I would approximate a globular protein as a sphere in 3 dimensions, and they are dissolved in water. And then the other category is fibrous or insoluble proteins. These are proteins like those that make up your hair, fingernails, skin, bones, teeth, etc. So these do not dissolve in water, and their jobs are structural in nature. Within these two categories of proteins, you have the same 4 levels of protein structure. The primary structure that we studied in the last module, which is the sequence, so it's characterized by the covalent connections from the N-terminus to the C-terminus of the polypeptide chain. Secondary structure, which is abbreviated 2 with the degree symbol, is based on local structures, and what characterizes secondary structure is the backbone hydrogen bonds. Think of those amide groups that can form hydrogen bonds. Importantly, the side chains are not involved in defining secondary structure. Tertiary structure takes into account those side chain groups, and this is the overall 3-dimensional shape and includes all weak forces, not just the hydrogen bonds. So ionic interactions, van der Waals, the hydrophobic effect comes into play, and of course hydrogen bonds. Quaternary structure is only for some proteins, those with multiple polypeptide chains or subunits, and they also pack together using all of the weak forces. All right, so the primary structure is shown here as an example. This is the amino acid structure of part of a polypeptide chain. So those are the covalent interactions between amino acids. Remember, primary structure is defined by the covalent interactions. Primary is up here now at the top in a smaller view, and now we're actually going to take the backbone of the primary structure, and we're going to wind it into this corkscrew shape. And it's now forming a secondary structure, in this case a helix. And the dotted lines indicate the backbone hydrogen bonds. So these are hydrogen bonds formed between the amide carbonyl group and the amide nitrogen's hydrogen in an amino acid above in the helix. And we'll go more in depth in this secondary structure in the upcoming slides. Now we're zooming out and we're looking at a sort of a, I don't know what, tube diagram of one subunit of the protein hemoglobin. And this is the beta chain of hemoglobin, which has alpha helices, which are the wider part of the tube representation shown here. And then connecting regions of the sequence are thinner. And then you go on to another alpha helix, etc. And these alpha helices dock against each other and with a heme group, a non-protein group that coordinates an iron to carry your oxygen in hemoglobin. But hemoglobin has quaternary structure. There are 4 polypeptide chains in functional hemoglobin. So that takes us to the quaternary structure. The quaternary structure, which is more properly pronounced quaternary, but my Hoosier accent comes out when I pronounce it as quaternary. And you can see that each one of the subunits of the hemoglobin is colored in a different color, and they look alike. In fact, the 2 alpha subunits fold into an almost identical structure to the 2 beta subunits, although they have slightly different primary structures. Okay, so we know about primary structure, and we're going to focus on secondary, tertiary, and quaternary structures, keeping in mind that these are held together by the weak forces that we talked about in one of the earlier modules of the course. We know that hydrogen bonds are going to be forming wherever possible, and those are short and straight. Remember, they're very directional. And they're especially important in secondary structure formation, which relies on those backbone amide groups to form hydrogen bonds. The hydrophobic effect is in effect, and it has to do with water and entropy, and that's one of the collapsing of the hydrophobic amino acid side chains into a protein's core is one of the driving forces for folding proteins into their 3-dimensional shape. Ionic interactions are abundant, especially on the surface of the proteins where they're soluble in water. Of course, that's opposite charges. And van der Waals packing is everywhere, the best packing, especially in the core of a protein.